Today we're going to talk about the cell cycle and the ability of cells to make copies of themselves. It is extremely important for cells to be able to create new cells in order to repair cells or for the organism to grow. At the beginning of the year, we talked about reproduction being a characteristic of life. We also talked about growth and development being characteristics of life. An organism grows when it adds new cells to the existing organisms. Cells must also be capable of getting larger. We're going to look at several different ways that cells divide, but today we're going to focus on asexual cell reproduction. We're going to look at several different types of cells and the way that they divide. First, we're going to look at bacteria, which divide via binary fission. Then we're going to look at somatic cells. Whenever you see the term somatic, you need to recall that somatic means body cells. These are cells like hair, nails, and skin. In another lesson, we're going to focus on the reproduction of sex cells, which are called gametes. Gametes are eggs and sperm. Those divide in a process called meiosis. We're not going to talk about meiosis today, just mitosis. If you recall, bacteria are prokaryotic cells. Prokaryotic cells do not have a membrane-bound nucleus. Bacterial cells have DNA that is in somewhat of a circular loop. When we look at binary fission, which is the way bacterial cells divide, the cell simply splits. What will happen is a new cell membrane is formed between the double DNA loop, and the cell membrane will pinch in two. It's a very simple process. When one bacterial cell divides, two is created, and bacteria divide really quickly. This is also a very efficient way of reproduction. Just like organisms have a life cycle, each individual cell has a life cycle. This is called the cell cycle. It starts or begins with interphase. The cell will spend the majority of its life in interphase. Interphase is made up of three parts. The first stage of interphase is called the gap 1 phase. It's often referred to as the first growth phase of interphase. This is where the cell grows and develops normally. It will get larger and replicate organelles. The second stage of interphase is called the synthesis stage. This is where DNA is replicated. It is important that DNA is replicated because the cell will divide and we need an original strand of DNA in one cell. We also need a copy of that DNA in the new cell. The next stage and the final stage of interphase is called the gap 2 phase. This is also known as the second growth phase. This is where even more organelles and protein is produced for the new cell. The next stage of the cell cycle is called mitosis. Mitosis is cell division. It occurs only in somatic cells or body cells. It includes 4 phases: prophase, metaphase, anaphase, and telophase, or PMAT. You will need to know each of those phases in order, how to spell them, and you need to recognize the cell if it is in one of those 4 phases. The last and final phase of the cell cycle is cytokinesis. Cytokinesis means division of the cytoplasm. Draw out the cell cycle and be sure to label each stage. We cannot talk about the cell cycle and mitosis without mentioning the chromosome. The chromosome is a structure that contains DNA. If you look at the diagram on the slide, you will see a large X-shaped structure. The center of the chromosome is called the centromere, and it holds each of the two halves together. Each of the two halves is called a chromatid or a sister chromatid. If we look down at the bottom of the chromosome, there's a structure called a telomere. It's very similar to an aclid or the piece of your shoestring that holds your shoestring together, that plastic piece that keeps it from unwinding. The telomere is the very end that helps to keep that DNA from unwinding. It keeps that chromosome in a solid structure. Chromosomes contain segments of DNA called genes. Different genes code for different features. Everything that you are is because of your genes. Sometimes these genes can work in groups, or sometimes they work individually. When we look at DNA structure, we've said that DNA is composed of nucleotides. It is a long strand of nucleotides. DNA is a supercoiled structure, and if you look at the structure of the chromosome, it contains supercoiled DNA. The DNA is supercoiled because it wraps around proteins called histones. So it's like tightly winding a string or strand around a tiny stone or ball, and that string will come out with a coiled shape, just like the DNA comes out with a coiled shape. This is how we can compact that DNA and keep it in such a small structure called a chromosome. Whenever DNA is not compacted or supercoiled in a structure called a chromosome, it's found as chromatin. So oftentimes you will hear me use the term chromatin in certain stages of mitosis. Here we're going to look at each individual stage of the cell cycle. First of all, it starts with interphase. In interphase, remember we said it's made of 3 stages. G1, S, which is synthesis, and G2. During this stage, the cell prepares to divide. If you look at the chromosomes, they are no longer in that compacted shape. They're actually in a stringy chromatin, so they have yet to get condensed into those thick chromosomes. So it's kind of a stringy chromatin. Also in this stage, you'll see that we have centrioles that have formed and then spindle fibers that have formed. These spindle fibers will move chromosomes around later on in mitosis. Mitosis is made up of 4 phases. This involves the division of body cells. The end result of mitosis will be 2 cells that are identical. Body cells are cells that have 2 sets of chromosomes. They get 1 set from each of their parents. So your skin cells, hair cells, bone cells, or nail cells have 2 sets of chromosomes. If we look at the first phase of mitosis, it's called prophase. During prophase, you can see that spindle fibers have formed and are coming from the centrioles. You can also see that the nuclear envelope has begun to dissolve, and you can see that the nuclear information is no longer in chromatin form. It is in condensed chromosomes. Metaphase is the second stage of mitosis. In metaphase, you can see that the chromosomes are now attached to spindle fibers at the centromere, and they have now moved to the center of the cell. You'll also realize there's no longer a clearly defined nucleus. Those chromosomes have left the nucleus to line up along the center of the cell. The third phase of mitosis is anaphase. In this phase, the chromosomes break at the centromere and move to opposite poles of the cell. This is the fourth stage of mitosis, and it's called telophase. You can see in telophase that this is the beginning of two new cells forming. You can see that the nuclei have begun to reform. You can also look at the chromosomes and see that they are uncoiling to return back to that chromatin structure. You can also see that there is a reduction in the length of the spindle fibers. Cytokinesis is the last stage of the cell cycle. This is where the cytoplasm divides equally between the two cells. If you look at the two cells, you can see that the cell has definitely divided into two separated cells. Both are genetically identical. This is why typically skin cells are all the same. Bone cells are all the same. This is the formation of two identical cells. If we look at the process in animal cells, remember animal cells don't have a cell wall. So in order for the cytoplasm to divide, there's basically a pinching inward. If you look at the cell, a pinching inward of the cell membrane, and that divides that cytoplasm equally. Now, if this was a plant cell, in the plant cell, we would have to form a new cell wall. So instead of a cell membrane pinching inward, we have a structure called a cell plate that's forming. These are vesicles that will eventually fuse together and become the new cell wall.